if you're gonna glaze over with this and you're checking for light leaks in your eyelids as we're going through, this is gonna be damaging because it's not gonna settle in and it's gonna be forever. Oh dang, what was I supposed to remember out there? Well, this is it, we're gonna propagate this. Notice there's a stimulus, boom! Something took place here. It changed the polarity from negative inside to positive inside, okay? But notice what happens. That is propagated, that whole process of the depolarization of the membrane in one action potential location triggers the opening of the voltage gated sodium channels in the adjacent portion of the plasma membrane. Which way is this signal going to go?
Down.
This way. It's going to be propagated this way. We're talking about this signaling that has to take place. The action potential propagates along the plasma membrane, along this action potential being propagated. Just as we propagated that paper lung. These people didn't move. That signal moved along here, okay? All right, so here's our NMJ, neuromuscular junction. AP, action potential, is coming down this axon right here. Now we're going to toss in another ion. Remember I told you that the more I study physiology, the more I understand how significant calcium ions are? Well, here we bring in a calcium ion. You probably can't see that from way in the back there, but this is a calcium, is a, carries a +2 charge on it. It's written as Ca as that calcium ion that we see here. Okay, so we have calcium ions have to come in here. Well, they're kind of important here. It says when an action potential reaches the presynaptic terminal. Are we good with presynaptic? If this area right here is called the synapse, and this is before the synapse, we're going to call it pre, and this is after the synapse, we're going to say it's postsynaptic. So pre and postsynaptic. It says here, presynaptic terminal of the motor neuron, it causes voltage-gated, here's voltage-gated sodium channels in the presynaptic membrane to open. As a result, calcium diffuses. Which way does calcium go? If it's going to diffuse, means what?
It's going to move down the concentration.
There's a whole bunch of it out here. There's not much in here. You open those channels, it's going to flood in there. It's going to go. When we hear diffusion, you have to know that means down the gradient here. Calcium ions, notice what the calcium does here. Real important job that it has in this propagation. It says calcium ions cause a few synaptic vesicles to migrate. The calcium promotes the movement of this over here to the end plate of this axon so that those are going to pop open like that. Have you heard the term exocytosis? Exo, something inside, exit, EX, exit, it's going to pop open. And now this chemical in here, in this case they're making reference to the acetylcholine, that chemical in there called a neurotransmitter has been released because there was an electrical signal. We're going to release a chemical here, so it's a chemical signal, and that's going to go across here, and it's going to then attach to a receptor here, and it's going to allow the movement of something across this open channel now, because that was a ligand-gated channel that required acetylcholine to activate it. So calcium ions cause these presynaptic terminal where they fuse with the plasma membrane. Okay, acetylcholine is released into the synaptic cleft. Exocytosis, EX, exiting. Acetylcholine diffuses. Are we good at that? Acetylcholine is going to diffuse. Where's it going to go? It's real high concentration here. It's going to diffuse across here to an area here where it can come in contact with a receptor. Binds to the ligand-gated. There's the term I asked you to remember earlier, ligand-gated. It just means he's got the right key. He can touch it and bam, we open that door and we can go across there. Ligand-gated sodium channels on the motor end plate, causing them to open. This is the motor end plate that we're talking about here. Sodium enters the muscle fiber. Sodium enters the muscle fiber, causing the postsynaptic membrane to depolarize. There it is again, we're going to depolarize. If depolarization passes threshold, remember about threshold? It had to be enough of a stimulus to trigger this next step, and action potential will be generated along the sarcolemma. Sarcolemma, remember what that term made reference to?
Plasma membrane.
Remember that? Sarcolemma, plasma membrane, same thing. Now, this is really important. Acetylcholine can't just camp out there. You can't have your ID and just leave it on the tab where you're supposed to tap it. Any of you use your credit card to pay for things by just tapping now? You got that little chip in there and you just tap it? What happens if you just leave it on there, parked on there? Well, hopefully it doesn't just keep tagging your account. Bunking, 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 55, 55, 55, 55. No, it just says tap. Doesn't it say tap? It doesn't say park your credit card right here. No, it says just tap it. And we're kind of told that that's the safest way to use your credit card today. Rather than slipping it in or sliding it down, just tap it. Going to Starbucks. Anybody here use Starbucks? My granddaughter works at Starbucks. She says, what, $13.50 for the mega, you know, what is it, Venti something or other, sprinkles, all the rest of it on there. And said people come in and do that every day. $13.50 for a, what is it, maybe about a 50-cent drink? Maybe $13.50, I don't know what it costs to make that. Tell me, what do you think it costs to make that drink? Ah!
$6.
$6 for yours, what is that? Is that a Venti?
Yeah, it's a cold brew with 3 pumps of brown sugar.
But is it from Starbucks?
Yeah,
It is $6.50?
$6.
You're
And I have
inexpensive. They don't want you coming
It's a
through
little
there real
less.
often. They want you to buy 3 of those at one time. Tap your card, right? You set
No, I use the app.
Oh. Is that different?
Yeah, because you have your card connected to
I
the
know,
app.
but is that different?
Not really, they just scan it. So-
Yeah, it's about the same thing, right? You're liganding, okay? Do you understand the ligand concept? The ligand means we have something here that can activate that. Yeah. And then it just immediately look at your account and says, whoa, only 2 minutes Starbucks this week. Acetylcholine detaches from the ligand-gated sodium channels, which then close. The acetylcholine does its job and then it has to get out of there. It can't stay parked on there. Notice this. Enzymes. Enzymes are real important. An enzyme here, acetylcholinesterase. When we have ASE after a particular term in the biological sciences, that usually makes reference to an enzyme. Put that down somewhere, park that away someplace. ASE after a term in the biological sciences usually means it's an enzyme for whatever that word is ahead of it, okay? What is it going to do? Acetylcholinesterase is going to do what? It's going to break down acetylcholine. And what will it break it down in? Into acetic acid and choline. Acetic acid is, well, you all probably have smelled acetic acid. It's like vinegar. Okay, acetic acid is a vinegar type substance. It breaks down acetylcholine to acetic acid and choline. Okay, so we have the ligand, which is the neurotransmitter here. It comes across, it parks on there, it opens the gate, does its job, but then it has to be dispersed. You can't leave it camped out there. Okay, so the motor neuron actively reabsorbs the choline in the axon terminal and is rejoined with the acetic acid. What is it going to do? We get back in here and it makes more acetylcholine in here. Recycling choline molecules require less energy and is more rapid than the continuously synthesizing. Easier to recombine that than it is to make new acetylcholine. Acetic acid is an intermediate in the process of glucose metabolism, which can be taken up and used by a variety of cells near, are we good with the term neuromuscular junction? You would describe, you should be able to describe the neuromuscular junction is where there's a communication between the neuron, axon of the neuron, and what? What is it? What's the word? Neuromuscular means what? It's between the neuron, the axon of the neuron, and the muscle. Neuromuscular junction should be decoded in your own mind right now. If you hear the term neuromuscular junction, you should be decoding that to say, oh, that's the communication between the neuron, the axon of the neuron here, and the muscle. Okay, so excitation, contraction, coupling is kind of interesting to me. It links the electrical and mechanical components together right here for what we're talking about. The action potential produced on the sarcolemma, an action potential on this outside of the cell right here, gets propagated, and it goes through something called these T-tubules. T-tubules are all of this little network of communication right in here. Calcium channels, or the sarcoplasmic reticulum open, calcium enters, and it binds to troponin. Have you heard the term troponin already? Does that sound familiar from the last time we met? Muscle contraction is going to take place here. So here's where we're, this small illustration right here. Excitation, contraction, coupling. Let's see if we can make them. Excitation. Something has to take place to fire this thing. Contraction. What does contraction mean? We're talking about muscles. What does contraction mean? Shortening, right? I mean, if you have talked to any pregnant females, they say, I'm having contractions. Is that a good thing? Well, depends. Okay? Are you at that stage in the delivery cycle here? Yeah, is it today? Yeah, it could be. I'm having contractions. What's that mean? The uterus says the bun in the oven is done. Okay? It's time to get out of here. All right. So, excitation, contraction, coupling. That is to say, we're linking some things. Begins at the NMJ, the neuromuscular junction, with the production of the action potential. What's the action potential? It has that boom across there in the sarcomere. The action potential is propagated. We go with that? Passed along the entire sarcolemma, along the entire surface of this, of the muscle fibers, and into this tubular mechanism here. They wrap around the sarcomeres. Are we good with the sarcomere? What did the sarcomere contain? Or at least two things that the sarcomere contained. What is it? What is it?
Actin and myosin.
You got it. You're exactly right. The actin and the myosin. The T-tube is wrapped around the sarcomere, where we find the actin, the contractile protein actin, and the contractile protein myosin, the thin filaments and the thick filaments, and they overlap and carry the action potential into the interior of the muscle fiber. There, the action potential causes voltage-gated sodium channels to open. When the calcium channels open, if calcium is sitting right outside the door and you open the channel there, what's going to happen? It's going to jump in there, right? It is opportunistic. It's opportunistic, which means what?
Take it if you got it.
It takes advantage of a situation. She wasn't watching her cup right there, so it just vanished, right?
Just took it.
I was opportunistic. I took advantage of the fact that she wasn't guarding her drinking mug here, okay? Do you follow what I'm talking about? If you open the channel, what's going to happen? If you've got water pressure in your pipes and you open the faucet, what happens?
So flooded.
Water comes out, right? Okay, think in terms of that. If you open the channel, it's not going to say, dang, I wonder what we should do now. No! Boom! Rapidly, notice this, rapidly diffuses. High concentration, low concentration, woo! Into the sarcoplasm surrounding the myofibrils. Once in the sarcoplasm, the calcium binds to what? Troponin. Troponin is this little nodule of protein substance, and its job is to open an area so we can have the actin binding head, or the sites there on the actin, open up. Binds to the troponin molecules of the actin myofilaments. The binding of the calcium to the troponin causes tropomyosin to move. Pull it up here. To move, which exposes the active sites on the actin myofilaments. The myosin heads then bind to the exposed, the myosin heads. The heads on the myosin are going to bind to the exposed active sites on the actin. Forget about the G there, to form the cross bridges. What's a cross bridge? It's where you have the myosin head attaches to the actin binding sites, and we now call that a cross bridge. The muscle contracts when the cross bridges move, okay? So, and here's what it looks like, cross bridge movement here. Here's our troponin right here. Calcium has to bind to that. This product right here is the tropomyosin. Tropomyosin usually sits right there blocking the actin sites. There's supposed to be a spot there, but the tropomyosin here is overlying that. This troponin has to pull this particular protein out of the way to expose that site so that this head of the myosin here has an exposed actin binding site here. The myosin head stores energy from the ATP, the myosin head. Here's a if we take ATP and we break it down, we end up with what? We take one away, what do we have? ADP. What does that mean? We have, let's decode this here. ATP, the T stands for what? Tri? What? Phosphate. What is the ADP? Diphosphate. And this can go down further, it can go to AMP. What's the M stand for? Mono. So you have ATP, which means that you have three phosphates here, is essentially, okay? ATP is, all right? You've got three phosphates. Well, you take one of them away, you've got diphosphate. You take two of them away, you've got monophosphate, okay? So this is ATP, this is ADP, this is AMP. We usually talk in terms of doing this. And all of you have rechargeable batteries, phones, computers, laptop, tablets, something like that, where what we're doing is we're constantly energizing. That is to say, we're taking that, whatever that nickel metal hydride or whatever it is you got there, that LiPo battery in there, and you go from one full charge and you knock it down, but then you have to plug it in to get it back up to full charge again. And that's what this does. We take ATP, it degrades to ADP, it gets sent back to where? Where does it get sent back to in order to get back to ATP? You should know this. What are you saying, Jade?
Aerobic cellular respiration.
Yeah, but there's a specific spot where it's sent to. What is that? Mighty mitochondria. Goes back to the mighty mitochondria because that's where this aerobic cellular respiration took place, within the cristae of the mitochondria here. All right, so the myosin head stores energy. The myosin head will remain in the high energy position until the muscle fiber is stimulated by a motor neuron initiating the event of excitation. So here's a cross bridge. Bam! That head is attached here is ADP, and there's the inorganic phosphate, cross bridge formation. Once the calcium binds, once it binds here, it pulls the tropomyosin out of the way so that these are exposed, and there's actually a location where it can hook up. The power stroke. Binding of the myosin head to the active sites on the actin forms the cross bridge and triggers a rapid movement of the myosin head. Kind of pulls. The movement of the myosin head is called the power stroke, because the myosin head is bound to the actin myofilament is pulled past. We're going to pull it past here, and then when we have the myosin myofilament, it is the actin myofilament that actually moves. Myosin stays put, actin moves, okay? The thin filament moves. Binding of ATP to the myosin head causes it to detach from the actin. Binding of the ATP, when ATP, boom, that causes that to detach. Constant. We only have a few seconds of stored ATP, which means what? Mitochondria have to continually be producing more ATP. You only have a few seconds of stored ATP. The myosin head breaks down ATP and ADP, and one of those inorganic phosphates remain attached. And so we have breakdown of the ATP. We have a recovery stroke. In the high energy position, the myosin myofilaments can be bind further down the actin myofilaments to another site, and it just kinda pulls its way along like that. Pull the actin myofilament even closer to the HL, further shortening the sarcomere. The cross bridge occurs when the myosin heads bind to the actin. Here's an actin site. That site on the actin wouldn't be available if tropomyosin were blocking that. And that's where you need the troponin with the calcium to open that up. During a single contraction, each myosin myofilament goes through the cycle of cross-bridge formation. It moves, pulls, releases, and then returns to its original position, and it's getting ready for the next boom. It starts all over again. Many cycles of power and recovery strokes occur during each muscle contraction. So this whole thing. So the summary of this contraction. An action potential is propagated along the motor neuron. Good at that? Propagated along the motor neuron. The action potential in the motor neuron stimulates the opening of the voltage-gated sodium channels. You got to get this real clear about what terminology we're using. And I'm putting it into proper perspective, because we talked about, you know, the resting memory potential and all that stuff. But right now, what we're dealing with is something which is this. And this is the MJ here. We'll talk about this whole thing right here. Go ahead, Mark. This whole thing right here is our NMJ, neuromuscular junction. Action potential is coming down this. This is what? What's the term we're using for this right here?
Down the grid.
Action potential.
The term we're using for this right here is axon, okay? Okay, it's part of the neuron. The neuron, the larger nerve cell up here, the neuron is up here, and this is the axon of the neuron. A little introduction to this. Understand this right now, and it'll make it a whole lot simpler. We have, here is our cell body, and we're gonna go down from here, and we're gonna have this thing like this. Okay, that's pretty good. This is Frank Netter, eat your heart out, okay? Dendrite. Cell body. Axon. Okay, and then this is gonna give rise to our neuromuscular junction. All right, when we talk about a neuron, we're gonna talk about dendrites, cell body, axon. What's the progression? How does it go? Dendrites, cell body, axon. Did you follow what I just said? Information comes in. This is like little antenna. Little antenna here getting information to the cell body, the neuron cell body. Dendrites, cell body, axon. Doesn't go the other way, it goes one way. What is the direction of passage? Dendrites. Follow along with me, don't get lost. This is real simple, three-step process. Dendrites, cell body, axon. Dendrites, cell body, axon. Dendrites, cell body, axon. Okay, got that? It's like something's in, something's out. Okay, this is dendrite, cell body, axon. So when we see this axon, that's what you're seeing right here. Where did this information come from? It came from some other impact right here. A thought came in and said, move that muscle. Contract that muscle. So what did it happen? Dendrites got some information processed through the cell body. What did they do? They sent it down the axon. Are you following me now? This axon just doesn't live out there like, oh, there's an axon. It's just hanging in the air like that. No! What is it connected to? The cell body, and then at some point, you know, the further backing this up, we say, well, there was a signal came in by way of a dendrite, and then it moved on down here. So this axon is a part of this drawing right here. So this axon is now, we're bringing the action, can do better than that. Action potential. Have you heard someone tell you that, oh, you have a lot of potential? What does that mean? You're a goof off? Loser?
You're great at- you can
What does
be
it
great
mean?
at
You
something.
got a lot of potential. What does it mean?
You can be good at something.
Man, hey, there's some talent here. It has potential, okay? It has the capability of doing something, of accomplishing something, okay? Of doing what? Creating some kind of action here. Are we following this? Very simple, very basic, very dumb explanation for this, but sometimes that's what we need to do is make it simplistic to clarify what's going on. The action potential is coming down. What kind of a signal is it?
Electric?
Let's try it again. What kind of a signal is this action potential?
Electrical.
There's a divided house here. Should we try again? Let's see if we can get a majority this time. The action potential is what kind of a signal?
Electrical.
Thank you. Does that sound right? Based on what we talked about before? It's an electrical signal. The action potential that's coming down here is electrical. Now, it's gonna come in contact with, as this action potential comes down, it opens some channels right here, right? As it gets down here, what kind of channels does it open? What are they?
Voltage gated channels.
Okay, but what kind of channels are being opened? Voltage gated gets part of the job done, but what kind of voltage gated are they?
Calcium.
Which ones are they?
Calcium.
Okay, we're getting some questions. No, what are you gonna say?
Calcium.
Calcium. Did you hear that declarative statement right up here in front? They were calcium. Remember, they're calcium. We have this calcium ion out here. And what does the calcium ion have to do? As this signal, this electrical signal comes down, man, who wrote that? ELPC. As the electrical signal comes down there, it kicks these gates open, and because there's a huge amount of calcium out there, what does it do? It sends one of these down to the edge right here. And what does it do? Exocytosis, okay? And what's the exocytosis gonna release?
Acetylcholine.
In this case here, we're gonna release a neurotransmitter. The one that we're gonna talk about when we talk about the muscle activity right here, the dominant one is acetylcholine. It could be any number of neurotransmitters. It just so happens that these right here are acetylcholine. So what's changed about this? We've gone from electrical signal, now we have what?
Chemical.
Thank you, we've got a chemical signal. So here's our synapse, our synaptic cleft, and the synaptic FT, our synaptic cleft. And then you know what? We've got a little ligand-gated channel right here that can open up and sodium can now rush in here. Okay? Any of that make any sense? And now what about if there's enough of a signal here, we say there's gonna be a what? Depolarization, remember that? If there's enough of a stimulus to trigger this, huh? Okay, so the action potential in the motor neuron stimulates the opening of the voltage-gated sodium channels. Exactly what Tim said up here. He's gonna take the stand here and do the rest of this lecture. Okay, so the influx of the calcium into the motor neuron causes into the motor neuron. We call this the motor neuron here, right? What does motor neuron lead you to think? What should you think when you hear motor neuron?
It facilitates movement.
It's gonna do what?
Facilitate movement.
Good answer, I like that one. Motor, it's gonna do something. Didn't say sensory neuron, it said motor neuron. Okay, remember the cranial nerves are motor and sense, wasn't there something like that? Yeah. Okay, so we're gonna have, and it's gonna cause the secretion of the release of the acetylcholine at that, right there, that vesicle is gonna rupture. The acetylcholine opens ligand-gated sodium channels in the sarcolemma. That's what happened here. It said, hey, open that gate. And now if there's an abundance of sodium right here, you open a gate, what's gonna happen? Boom! It's gonna go down the concentration gradient. The resulting action potential in the muscle fiber travels along the entire sarcolemma. Action potentials also move down the T-tubules. T-tubules wrap around the sarcomere. Remember the sarcomeres? Actin and myosin. Where actin and myosin overlap and carry the action potentials into the interior of the muscle. There the action potentials, the APs, cause gated channels in the terminal cisterna. Again, more calcium. When the calcium channel is open, calcium rapidly diffuses. Remember, we need the calcium here because we have tropomyosin and troponin, right? And we have to make the actin binding sites available. Once in the sarcoplasm, the calcium binds to the troponin, on the actin. Binding of the calcium troponin causes tropomyosin. Move! Poof! Exposes the sites for the actin. Myosin heads are then capable of binding, forming the cross bridges. Cross bridge was actin, myosin. Muscle contracts, and the cross bridges move. ATP, got that? Adenosine triphosphate molecules are broken down into ADP, and this inorganic phosphate releases energy needed to move the myosin head. Boom! The heads of the myosin myofilaments bend, causing the actin to slide past the myosin. Sliding filament theory. As long as calcium is present, as long as there's enough calcium there, the cycle's going to repeat. 3 major ATP dependent. That sounds like one of those questions, doesn't it? 3 major ATP dependent events are required for muscle relaxation. After an action potential has occurred in the muscle fiber, the sodium potassium pump must actively transport sodium out of the muscle. Yes? Remember that one? ATP. Sodium potassium pump. 2. ATP is required to detach the myosin heads from the active sites on the recovery stroke. 2. Third one, 3 major ATP. ATP is needed for the active transport of calcium into the sarcoplasm, particularly for the sarcoplasm. 3 ATP dependent events here. Oh, let's see. Let's do this. Give me just a moment to bring this other screen up, and we'll see if we can. Don't want to blast everybody out, but let's try this.
Basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting